Welcome to the Deep Dive. Today we're cracking open one of the most fundamental ideas in all of medicine, but one that had, well, a surprisingly rocky start. The drug receptor concept.
It really is the invisible handshake, isn't it?
Yeah.
It dictates how almost every medication works. When we talk about receptors, what we mean are these protein structures.
Right, they can be on the surface of a cell or even inside it.
Exactly. And they mediate the effects of chemical messengers, hormones, and of course, pretty much any therapeutic drug you'll use in practice.
Absolutely. And for anyone studying pharmacology or medicine, this idea is just bedrock. But here's the thing that's so fascinating, the historical twist.
Oh, yeah.
This concept, which seems so obvious to us now, actually spent nearly a century in a sort of scientific purgatory. It was highly contested, a lot of people ignored it, and it didn't really get fully accepted until the 1960s.
So our mission today is to trace that whole journey. We're going to go from, ancient philosophical ideas all the way to modern molecular cloning.
And to really get why this was such a big deal, we have to start at the beginning, when pharmacology was, frankly, a bit of a mess.
A huge mess. Treatment was often random, sometimes harmful. You had the Hippocratic humoral theory dictating everything.
The four humors: blood, phlegm, yellow bile, black bile.
Precisely. And the whole therapeutic principle was contrarius curantur.
Opposites are cured by opposites.
Right, so if you have too much cold phlegm, you get a hot treatment. There was just no concept of molecular specificity at all.
But there were these tiny glimmers. If you dig way back, the Epicureans had this idea about substances becoming, and this is a quote, entangled or interlocked through minute hook-like extremities.
Which is just wild to think about. That's 2,000 years before anyone even knew what a protein was. It's the very, very first intellectual seed of our modern lock-and-key model.
So how did we get from that ancient speculation to something more scientific?
Well, things started to shift in the 19th century. Science moved its focus from whole-body humors to the cell itself. Rudolf Virchow's cellular pathology in 1858 was a huge part of that.
So disease was now centered in the cell, which meant pharmacologists had to start thinking about specific cellular targets.
And they were already seeing it in action, even if they couldn't explain it. People noticed that strychnine seemed to have an elective affinity for the spinal cord. Certain toxins only hit certain tissues.
So from that idea, elective affinity, to a real formal receptor theory, who made that jump?
That brings us to Paul Ehrlich around 1900. And what's crucial to remember is that Ehrlich wasn't a pharmacologist.
He came from immunology, right?
An immunologist and a toxicologist. He developed what he called the side chain theory to explain how cells dealt with bacterial toxins.
Okay, so let's unpack his terms, because they're a little unique to his field.
Absolutely. He proposed cells had these side chains that could bind to toxins or immune bodies, basically neutralizing them. And he's the one who coined the term receptor to describe the central role of these side chains.
He had different types too, didn't he?
He did. He had things like amboceptors, which were his idea of a complex receptor with two binding groups, and sessile receptors, which were the ones that stayed put fixed to the cell surface instead of being released.
So he's really mapping out the cell's immune hardware. And he was practical too. He famously used his knowledge of chemical staining to look for magic bullets.
Yeah, his thinking was if a dye sticks to a specific cell, maybe a drug based on that dye could kill that cell. And that line of thinking eventually led to salvarsan, the first real modern chemotherapeutic.
A huge step, but as you said, still rooted in immunology. The big shift to drugs specifically came from someone else.
It did. John Newport Langley, a physiologist at Cambridge in 1905. He's really the first person to propose a full receptor theory for how drugs and neurotransmitters work. He called them receptive substances.
And Langley backed it up with some really elegant experiments.
He did. He was looking at antagonistic drugs like pilocarpine and atropine on glands and muscles. But his killer experiment involved nicotine and curare on sympathetic ganglia.
Okay, what was so important about that one?
He showed that the drugs worked directly on the muscle cells, even if the nerve endings were gone. So he concluded that something like curare must form a reversible curare muscle compound by chemically uniting with a receptive substance.
So it wasn't just blocking something mechanically. It was a true chemical union at the target.
That was the massive conceptual leap.
So by 1905, the chemical theory is on the table. It's got strong evidence. So why wasn't that the end of it? Why did it face so much resistance?
because it just flew in the face of the whole scientific mindset of the time, that 19th century positivism. Scientists wanted to measure physical constants they could see and quantify, not speculate about invisible proteins.
So they came up with competing physical theories.
Exactly. The idea that it was more like, say, oil and water, they argued it all came down to stuff you could measure, like solubility, surface tension, concentration.
Who were the main players on that side?
Well, a major one was Walther Straub. He developed the poison potential theory. His idea was that a drug's effect depended entirely on the difference in concentration between the outside and the inside of the cell.
So for him, it wasn't about binding. It was about the drug's physical potential to get across the membrane. The key was solubility in the cell wall.
Precisely. And there were others like Isidor Traub and his theory of attaching pressure, which focused on surface activity. This was actually pretty good at explaining why some drugs were so potent in tiny doses.
Because they could disperse easily across the membrane.
Right. And even guys like Arthur Robertson Cushnie, who sort of accepted some kind of attachment, still argued that the effect was mainly determined by physical things like volatility or solubility.
This sounds like a complete scientific stalemate.
It was. And to make it even harder for the receptor theory, another hugely successful field of research came along that let scientists just, well, sidestep the whole question.
You're talking about the transmitter theory.
Yep. Henry Hallett Dale and Otto Louis, who shared the Nobel Prize in '36 for it, they were brilliant. They focused on isolating the chemical messengers themselves, like acetylcholine.
And they measured what those chemicals did.
Which was incredibly valuable, but they effectively postponed solving the how. They didn't need to know how acetylcholine reacted with the cell, just that it did. So for decades, the receptor was this ghost, fighting physical theories and being ignored by the most successful neuropharmacologists.
Okay, so the receptor concept needed a lifeline. And you're saying that lifeline came from math.
It did. We have to jump forward to Alfred Joseph Clark in the 1920s and 30s. Clark was the one who gave the theory a rigorous quantitative foundation. He took Langley's idea of a chemical union and turned it into a mathematical model.
So what did his model show?
It showed that drug action was a reversible chemical combination, and crucially, that the effect was proportional to the number of drug receptor complexes that were formed.
And he could use that math to challenge the physical theories, like Straub's concentration idea.
Yes. He did this amazing work on competitive antagonism, using acetylcholine and atropine on a frog heart. Clark showed that atropine kept working even after he'd washed it out of the system.
So that lingering effect couldn't be explained by a simple concentration gradient.
Exactly. It could only be explained if the atropine molecules were actually fixed to the cell surface. It was a clear quantitative win for the binding theory.
Okay, so Clark establishes the core idea. Effect is proportional to occupancy. But that simple model couldn't explain everything, could it?
No, it couldn't. It didn't explain the difference between a drug that produces a massive effect and one that produces only a small one.
A full agonist versus a partial agonist.
Right. So the theory needed refining. And that massive leap forward came from R.P. Stevenson in 1956.
Stevenson is the one who gives us our modern understanding with two critical new concepts.
Efficacy and spare receptors.
Let's talk about efficacy first. That's the game changer.
It's the whole game. Clark had focused on affinity, basically, how well the drug sticks to the receptor. Stevenson said that's only half the story. Efficacy is what happens after the drug binds. It's the drug's ability to actually flip the switch and initiate a response.
So you could have two drugs that stick equally well, same affinity, but one is a full agonist with high efficacy, and the other is a partial agonist with low efficacy.
You've got it. And then he added the idea of spare receptors.
Which explains why you don't always need to occupy every single receptor to get a maximum response.
Right. If a cell has 100 switches, but you only need to flip 10 of them to get full power, the other 90 are spare receptors. It gives the system sensitivity and amplification.
So we have the math, but we still need a way to classify these things.
And that's where Raymond P. Ahlquist comes in 1948. He took a bunch of adrenergic substances, adrenaline, noradrenaline, and so on, and tested them across 23 different tissues.
And he saw different patterns of response.
He did. He saw clear, distinct potency rankings, and that led him to propose the dual adrenoceptor theory.
The existence of alpha and beta receptors.
Yep. He saw that alpha functions were mostly about excitation, like vasoconstriction, while beta functions were more about things like vasodilation and exciting the heart muscle.
But at this point, this is still a theoretical classification, right? It's based on potency rankings. It's not a physical thing yet.
Totally theoretical. But it's the theory that led directly to the moment the receptor became undeniable, the clinical validation.
Sir James Black.
So James Black. He was trying to treat angina and hypertension, and he later said he was directly inspired by Alquist's paper. He thought, if Alquist is right, if there really are separate beta receptors on the heart,
Then maybe we can design a drug to selectively block them.
and that's exactly what he did. His team's work led directly to the development of the first beta blockers, like propranolol, in the 1960s.
And they worked.
They worked perfectly. They did exactly what Alquist's theory predicted they would. And when you have a drug that successfully treats massive diseases like hypertension, the entire medical and scientific world has to sit up and take notice. The receptor was no longer a ghost. It was a real, actionable target.
A stunning moment. The receptor is now clinically proven. But even then, in the 60s, no one had actually seen one. No one had isolated this protein complex.
Right. The final frontier was to actually get your hands on it, and that was an immense challenge. These things exist in tiny, tiny amounts in the cell membrane.
So how do they solve that?
First, they needed a source that was just packed with receptors, and they found one in marine biology, of all places.
The electric organs of fish.
The electric organs of the torpedo ray and the electrophorus electric eel. They're basically just densely packed synapses, super rich in nicotinic acetylcholine receptors.
Okay, so you have the source. Now you need a way to tag the protein you're looking for, a chemical beacon.
And nature provided that too in the form of a very specific snake venom, abungarotoxin. It binds so tightly and so specifically to that receptor that it was the perfect tool to track it and purify it.
And with those tools, researchers like Jean-Pierre Changeux and Riccardo Maletti in the early 70s, they finally did it. They isolated the nicotinic acetylcholine receptor.
The ghost was finally a molecular reality. They proved it was a distinct protein complex.
And then came the final piece of the puzzle, the blueprint.
The blueprint. Shisaku Numa and his team in Japan between 1982 and 1983, they successfully cloned the genes for all 4 subunits of that receptor.
And cloning it did more than just identify it. It confirmed its fundamental structure.
It did. It showed these subunits came from a common ancestor through gene duplication, which is the basis for how we define entire receptor families today. And it immediately opened the door to understanding diseases caused by receptor dysfunction, like myasthenia gravis.
So, to recap this incredible journey for everyone listening, the receptor starts as a philosophical idea. It's given a chemical basis by Ehrlich and Langley. It nearly gets killed off by physical theories. Then it's rescued by the math of Clark and Stevenson.
Then it's functionally proven by the clinical success of beta blockers inspired by Alquist's classification.
And finally, it's confirmed as a physical molecular reality by the cloning work of Numa's team.
It's just a fantastic story of scientific persistence, crossing all these different disciplines: physiology, math, toxicology, and finally molecular genetics.
Okay, so for everyone following along, here's something to think about to really cement these concepts. We discussed how Clark's initial theory focused mainly on a drug's affinity for the receptor.
How well it sticks.
Right. Now, I want you to explain why Stevenson's later introduction of the concept of efficacy was so essential for explaining the diversity of drug actions we see, especially for explaining the existence of partial agonists.
Think about what each term is actually measuring. One is about sticking to the lock. The other is about actually having the power to turn the key. That's the crucial difference.